podcast number 9. And in this podcast, we're going to give an example of how genes are regulated to control RNA production, and then start to think about sorts of questions that you would get about gene regulation. So the idea is, I said in an earlier podcast, that every single cell has all of the instructions to make a whole person. Every single cell has all the DNA to make a whole person, but every single cell doesn't transcribe or translate all of that DNA at the same time. So you have nose cells that have all the DNA to make a person, but they are making transcribing nose genes and then translating those transcripts, RNA transcripts, into nose proteins. And then you also have eye cells that are transcribing eye genes and then translating those transcripts into eye proteins. It's not that every cell makes everything. And not only at one place in time, but over time, organisms change their gene expression with development. So this silly picture of a toddler with a beard, that looks very strange because toddlers don't make beards, even though they have the genes to make a beard. That happens later on in development, hopefully much later, when they're adults. And it is as a result of the response to environment, changes with developmental age, etc. So gene expression changes over time. How is it that organisms respond to their environment? How is it that organisms select which genes to express under which conditions? And a way to understand this has come from the study of what's called bacterial operons. So this is the idea that there are sets of genes that can respond to changes in the environment, and this is in general called gene regulation. A model for understanding how this works is the bacterial operon model. A model is just a way of understanding it, often much more simple than the real life situation, just so that we can understand the basic principles of what's going on. And then when we get to the real situation, we can add complexity to that model. But to understand the basics, two French scientists, Jacob and Monod, worked with bacterial systems. Bacterial systems are much less complicated in gene regulation than eukaryotic systems, in other words, but they still offer lots to understand the basic ideas of how things work. So in a biochemical pathway, and we will study a couple biochemical pathways in detail, you have whatever molecule starts, and then protein enzymes work on that molecule, change the shape of that molecule into something else, and then there is a series of these changes over time until the desired molecule is produced. And there's a common phenomenon that there is feedback of the concentration of this desired molecule will interact with an early enzyme in the pathway, such that if there's a lot of this molecule made, that gives information back to the beginning of the pathway to shut down this pathway. If you think of like an assembly line, if there's a lot of widgets coming off the end of that assembly line, there's feedback to the beginning of the assembly line in order to shut down. We don't need so many widgets anymore. And this happens in cells also, that if a certain thing is produced, it's produced to a certain concentration until it's not needed anymore. It's not that you flip the switch on and it stays on forever. And in fact, when this gene regulation goes haywire, where things are switched on and they stay on, or switched off and they stay off, the inability to regulate leads to disease, such as cancer. So, the idea is that there are genes that are related to each other, that they do similar things, and they are physically next to each other in a bacterial chromosomes. Bacterial chromosomes have a different shape than eukaryotic chromosomes. They're circular instead of in a line, and they tend to be simpler in how they're organized in terms of eukaryote versus eukaryotic systems. And these cluster of genes are related to each other in terms of what they do. They're controlled by a single switch that turns everybody on or everybody off downstream of that switch. The switch itself is a piece of DNA called an operator, and that operator is within a control element of the DNA called a promoter. So both bacteria and eukaryotes have promoters. Promoters in general are receiving signals from the cell saying, should I make this thing and how much should I make? It's not, it's receiving that information, and then it passes that information on to the operator. The RNA polymerase that's going to read the DNA and make mRNA, and then that mRNA will be translated into protein. That RNA polymerase binds at the promoter. So the promoter doesn't make the protein, but it's an important starting point for making a protein. And the operator is the actual on-off switch that is within that promoter. An operon sounds similar to operator, but it's a different thing. An operon is the whole system, so the entire stretch of DNA that includes the operator, the promoter, and all the genes that they're controlling. So it would be the wiring, the light switch, and the lights. The whole thing is the operon. Okay. The thing that turns on or off the operon is a repressor, and it's a protein. The protein is made all the time, and the repressor decides whether or not it's going to switch the operon on or off depending on some signal it's going to get from the environment. And hopefully this will become obvious in a second. Okay, but when it is repressing, it is physically binding to the DNA and blocking RNA polymerase from getting past the promoter. So RNA polymerase binds to the promoter. The operator is downstream of that. It has something bound to it, the repressor protein, and so physically the RNA polymerase can't get past that in order to transcribe RNA. The repressor is made from a gene that is outside of the operon, and that gene is always making the repressor protein. That's called a regulatory gene. So how does the RNA polymerase block the operon? This is the repressor protein here. I said RNA polymerase blocked. RNA polymerase is blocked by the repressor protein. This thing in green here has 4 different subunits, light green and dark green, and it binds to the DNA. That's this molecule here, that is a loop, and it binds to that DNA, and then RNA polymerase, which is binding upstream here, physically can't get past that. The genes you want to transcribe are downstream here, and RNA polymerase can't physically get past it. So that's how it turns on or off the switch. The things that the operon are going to make are genes that are going to break down break down a sugar called lactose. So the operon we're going to talk about is called a lac operon, lac for lactose. Lactose is a combination of 2 sugars, galactose and glucose, and the gene that is made by the operon will break apart lactose into galactose and glucose, and then the bacteria can eat those two things. Okay, so that's why it is turning on the operon, is that it wants to eat. There's lactose in the environment. It wants to break down lactose into galactose and glucose so it can get energy from it. Okay. The lac operon contains genes that code for enzymes used in the hydrolysis and metabolism of lactose. That's the beta-galactosidase that I was just talking about. Okay, the gene that gets transcribed and then translated into this protein will break down lactose only if it's around. Okay, hydrolysis just means splitting. The lac repressor protein is active and switches the lac operon off. That's this green molecule I was talking about before. That's a protein that's going to bind to the DNA and prevent the RNA polymerase from transcribing those genes. And a molecule called an inducer inactivates the repressor to turn the lac operon on in the sense that it will translate or transcribe proteins. In the case of the lac operon, lactose itself, which is a carbohydrate, that's sugar, is going to be the inducer. It's going to bind to the repressor protein. It's going to cause the repressor protein to change shape. And then when that repressor protein changes shape, it'll fall off the operon and allow RNA polymerase to do its thing. So we have a situation where we're trying to figure out how does an organism decide when it should make a gene and when it should not make a gene. And by say make a gene, I mean transcribe that genetic information into RNA and then translate that RNA into protein. The organism would not want to do this just all the time because it's expensive. It's expensive in terms of energy to make RNA, and it's expensive in terms of energy to turn that RNA into protein. So if there was nothing to eat, you wouldn't want to make the stuff to break down stuff to eat because that would cost you energy, and the whole thing you're after is energy. So you only want to turn on this system when there is something to eat. And so they are listening to the environment, and they have a way of detecting whether or not lactose is around, and then turning on the machinery to eat it. And then when lactose goes away, they have a way of discovering lactose is not around anymore and shutting down the factory. All right, so operons are typically drawn as like a bar diagram like this. And this would be the 5' end, this would be the 3' end. This bar here is DNA, it is bacterial DNA. And this is just sequences of A's, C's, G's, and T's next to each other. For a stretch of sequence, that's called the promoter. That's where RNA polymerase is going to bind to the promoter. The repressor protein is going to bind to the operator. And then these genes, like Z, like Y, like A, one of these is the gene for beta-galactosidase. Another one is a gene that makes it easier for lactose to get into the bacterial cell. And the third one we don't talk about because it doesn't really do much in the sense of what we're talking about here. Okay, so this repressor protein is bound to the operator. RNA polymerase is bound to the promoter, and it can't get physically past that operator. It needs to read these genes in order to make the protein. So again, the repressor is a protein. This is what it looks like when it's bound to the DNA. And then the RNA polymerase can't get past this, physically can't get past this, and so you don't make genes Z, Y, and A. So the repressor protein is made all the time, and if there's no lactose around, the repressor protein is always bound to the operator. Okay? Then RNA polymerase, either if it's bound to the promoter or not, doesn't matter, it can't get past the repressor, and this is quote-unquote good for the bacterium because if there's no lactose around, you don't want to transcribe these genes and then translate those mRNAs because that would cost a lot of energy and there's no lactose around to eat, so no way to get energy. Okay? So the repressor protein represses the operator. When lactose is around, lactose gets converted to another chemical form called allolactose. It binds to the repressor and it causes the repressor protein to fall off. Now RNA polymerase can bind to the promoter. It can get past the operator because there's no repressor protein there, and it can go ahead and transcribe Z, Y, and A. That mRNA gets transcribed into mRNA for Z, Y, and A, and then it gets translated into protein, and it makes beta-galactosidase, which can break down the lactose. Why would you do it in this situation? Well, we know lactose is around because lactose is bound to the repressor protein, knocking it off the operator. And if lactose is around to bind to the repressor protein, it obviously is around to be eaten. And so you make the enzymes to break down lactose and get energy from it. Eventually, you'll run out of lactose. You'll chew up, the bacterial cell will chew up all the lactose, break all those bonds with beta-galactosidase, including the lactose bound to the repressor protein. The repressor protein is no longer repressed. It goes back onto the operator, and you shut the whole business down when you run out of lactose. So lactose is the inducer. If lactose is around, RNA polymerase makes it past the operator. LAC-Z, LAC-Y, and LAC-A are transcribed. These are protein coding genes. They make enzymes that break down lactose, like beta-galactosidase, and then the cell can get energy from breaking down that lactose. Okay, so that is one way of getting introduced to lac operons. Lac operons, again, as a model for how an organism can respond to the environment and decide when it's going to transcribe genes and then translate those mRNA transcripts, and then when it's not going to. And it doesn't make any sense for the cell to make the stuff to break down lactose if there's no lactose around. So if there's no lactose around, the repressor protein blocks RNA polymerase from transcribing those genes, and they're not transcribed, and you don't break down lactose because there's no lactose around. If there is lactose around, lactose is the thing that knocks the repressor protein off the operator, and then the RNA polymerase can get past the operator, transcribe genes Z, Y, and A, translate those into proteins that break down lactose. Once all the lactose is broken down, then now you're out of lactose, and again the repressor protein goes back on the operator and shuts it off. Okay, so that's a simple way of looking at lac operons, but they're actually more complicated than that. And just like you, bacteria have a preference for some foods over others. So maybe you choose Pop-Tarts over kale, for example. So if glucose is the version of Pop-Tarts, it's preferred over lactose by bacteria. If glucose is in the environment, they much rather break that down than to break down lactose first. So if it has a choice between glucose and lactose, they will not turn on the lactose operon because they'd much rather use glucose. The lactose operon only deals with lactose. There's other genes that get turned on for the presence of glucose. So the lactose operon, we're only dealing with that, and it's just making a decision, should I make the stuff that break down lactose? And if glucose is in the environment, the answer is no. Because if glucose is around, that's what I want to eat. If I can have Pop-Tarts, that's what I want. Okay? If there's no Pop-Tarts available, and there is kale available, well, you got to eat something. So then you eat the kale. Okay? So, how is glucose sensed by the lac operon? It's sensed by a protein called the cyclic AMP response protein. CAMP response protein, or CRP for short. Okay? When glucose is high, cyclic AMP is low. When glucose is low, cyclic AMP is high. So cyclic AMP is the opposite of whatever glucose is. All right, so remember that. Now, cyclic AMP responsive protein, or CAP, or CRP, binds to the promoter, and it has to be on the promoter for RNA polymerase to be bound to the promoter. So we didn't show you this before because it's just trying to introduce the idea of how operons work, but this is how they really work. In that first you have to have cyclic AMP and CAP bound to the promoter before RNA polymerase can bind to the promoter. And if that's the situation, then RNA polymerase can bind to the promoter, and depending on what the situation is on the operator, it can either get past the operator or not get past the operator. Okay, so cyclic AMP needs to bind to CAP in order for CAP to bind to the promoter. Cyclic AMP is the opposite of glucose. So cyclic AMP is high, glucose is low. So if that means no glucose is around. And then if no glucose is around, then I might want to break down lactose because there's no glucose around. No Pop-Tarts around. I might want to eat some kale. If cyclic AMP is low, that means glucose is high. Remember, cyclic AMP and glucose are opposites. If cyclic AMP is low, glucose is high. If cyclic AMP is low, then the CAP protein can't bind to the promoter, cannot bind to the promoter, and then that means RNA polymerase can't bind to the promoter. But that's okay, because cyclic AMP is low because glucose is high, and I'd much rather eat glucose anyway. So you would not want this operon to be on in the case of cyclic AMP is low and glucose is high. Okay, because glucose is high, you want to eat glucose. You only want RNA polymerase to bind to the promoter if there's no glucose around. If there's no glucose around, cyclic AMP is high. Cyclic AMP binds to the CAP protein. That binds to the promoter. Now RNA polymerase can bind to the promoter. Okay? So the cyclic AMP concentration is opposite the glucose concentration. Cyclic AMP is high, glucose is low. When cyclic AMP is low, glucose is high. Cyclic AMP is low, then this would not happen because you need cyclic AMP for the CAP to bind to the promoter. And if CAP's not bound to the promoter, RNA polymerase is not bound to the promoter, and this is off. Okay? So CAP only binds to the promoter with cyclic AMP. That only happens when glucose is low and cyclic AMP is high. And then if glucose is low and lactose is low, well, it doesn't matter if this thing is bound or not because it's not going to get past the repressor. If glucose is low and lactose is high, now lactose will bind to the repressor protein falls off, and then RNA polymerase can go ahead and make the enzymes to break down lactose. So once again, the lactose operon only deals with lactose. It can sense whether or not glucose is around, but it's not going to make the stuff to break down glucose. That happens someplace else. The only decision that this operon is making is, should I make the stuff to break down lactose? And the answer is no if there's a lot of glucose around. The answer is no if there's no lactose around. The answer is only yes if there's no glucose around and there is lactose around. Okay. If there's glucose around, you do not want to turn on the lac operon. Cyclic AMP was low, CAP does not bind to the promoter, and no transcription of LAC, Z, Y, and A. And those are the genes to break down and import lactose. If there's no glucose around, then you do want to turn on the lac operon, but only if there's lactose around. So no glucose around, and maybe there's no lactose around. It doesn't make sense to make the stuff to break down lactose because there's no lactose around. So everything is shut down. But if there is no glucose around and there is lactose around, then you want to turn on the lac operon because that'll break down lactose and give the cell some energy. If both glucose and lactose are around, you again don't want to turn on the lac operon because the cell would rather eat glucose than eat lactose. And so if glucose is high, that means cyclic AMP is low, and the cyclic AMP response protein will not bind to the promoter, and therefore you can't turn on the operon. All right. If we're talking about eukaryotes, so that probably seems really complicated if you haven't heard about this stuff before. If we are talking about eukaryotes, things with a nucleus, it is even way more complicated. So in eukaryotes, you have transcription and translation physically separated from each other. Transcription happens in the nucleus, and translation happens out in the cytoplasm. In bacteria, they don't have a nucleus, so they can happen all in the same place. There's all kinds of regulation besides the sort of stuff that I just talked about. It happens at many levels. The first level begins of control of access to the DNA. This is how the DNA is wrapped. You have to unwrap the DNA in order to do transcription, and how the DNA is wrapped is affected by something called epigenetic regulation, and that can affect whether or not you turn on a gene. And then instead of a single repressor protein binding to the operator, you have many, many proteins, dozens of proteins that are binding to the DNA and controlling whether or not that gene gets transcribed. So it's lots more complicated. Here is an example of a promoter in a eukaryotic gene. Here are some of the many proteins that are assembled on that promoter before that promoter can release RNA polymerase to transcribe those genes. Those many proteins end up binding DNA that is bent around and is very far away, maybe as far as 50,000 base pairs away from the part that actually turns into protein. Any of these things can affect whether or not RNA polymerase gets to do its job. So it's lots more complicated. Okay? Any kind of mutation that affects the size of this loop, like a big deletion or a big insertion of DNA, can also affect whether or not you get to make those proteins. So the bottom line is eukaryotes are much more complicated, but we can get an idea of how organisms respond to their environment, like is there lactose around or not, and I would want to make a translated gene in this situation, and I would not in that situation. We can get an idea of how that works in bacteria.